It looks like we are streaming on the YouTube, and I just want to welcome everybody today to the Hawaii Invasive Species Council Brown Bag Series, which are short talks with invasive species experts and policymakers from across Hawaii. We do these talks to help share what's happening in invasive species work statewide and help make connections with between science management and community. So, as I mentioned, today's session is part of Stop the Ant Month, and we're lucky to be joined by Monte Tudor-Long and Trisha Dillenburg from Dillenburg, from the Maui Invasive Species Committee. Monte will share what more than 50,000 ant detections reveal about ant diversity on Maui. And Trisha will take us behind the scenes of MISC's Little Fire Ant Detection Dog Program. So we also have today, we're getting some help from behind the scenes from Chuck Camara, who is another one of the HISC support staff. He is the Hawaii Pacific Weed Risk Assessment and Plant Pono Specialist. Mahalo, Chuck, for helping. And aloha, I didn't introduce myself. My name is Elizabeth Spieth. I'm one of the HISC support staff and the manager of the state pest hotline 643pest.org. So before we get started, I also wanted to do just a quick shout out. We have another HISC Brown Bag that'll feature You Heroes Biocontrol Economics Team. So they're the folks at the University of Hawaii that do economic analysis of all kinds of things. And they're going to present on Wednesday, November 5th, diving into the cost and benefits of biological control programs in Hawaii. And Chuck shared a link if you want to read a little more or register for that brown bag talk. So once again, mahalo for joining us today. And with that, I am going to tell you a little bit about Monte Tudor-Long, who will be starting off today's talks. So Monte Tudor-Long has been with the Maui Invasive Species Committee since 2015 and was a founding member of the MISC Little Fire Ant crew. He is currently MISC's invasive ant early detection and rapid response technician. He holds a bachelor's degree in environmental studies from the University of California at Santa Cruz. And he's especially interested in the history of changes to Hawaii's biodiversity, studying the sequences of extinctions and the arrival of invasive species over time. His coworkers call him Uncle Ant. And with that, I'm going to hand it over. Oh, and if you don't mind, please, if you have any questions, enter them into the Q&A, which is a box at the bottom of your Zoom. And we will address those either live through typing in your answers or answering questions at the end of this overall session. And with that, I'm going to hand it over to Uncle Ant.
All right, thank you so much, Beth. I really appreciate that introduction. And so once again, my name is Monte. I work at the Maui Invasive Species Committee. I am the person at MISC who identifies all the ants that we collect. And you are the people who collect those ants, or you're people who see ants. You're people who encounter ants because you're just a human living on Maui, or you work for an agency or a company where people come to you and ask you about the ants they're encountering. And this talk is about not just little fire ants. Little fire ants are a very important part of the invasive ant situation in Hawaii, but I want to talk about invasive ants more in general. I want people to think more than just whether an ant is a little fire ant or not a little fire ant. We call that a presence-absence situation. Yes, it's LFA, or no, it's LFA. I want people to collect an ant, turn it in, and say, what is this ant? What is this ant doing on my island, and what can I do to get rid of it? Or should I even care about getting rid of it? So this talk is going to cover a lot of stuff about all the different ants in Hawaii and when they got here and what they're doing here and what's coming around the corner. I am trying to advance my slide and nothing is happening. Let's see. Oh, I think I got it. So History of ants in Hawaii is a big topic of what I'm going to talk about here. I want to talk about when ants arrived and what ants are here today. I want to talk about this because we need to know what all is out there. The ants in Hawaii all interact with each other. They all interact with our environment. It's not just little fire ants. All the ants in Hawaii are part of our ecosystem. I also want to touch on the concept of ant identification. In the past, there was a lot of effort, as I said, on just presence, absence. Is it little fire ant? Is it not little fire ant? I want us to move towards a situation where we're actually identifying all the ants that are collected so that we have a better picture of what is happening in our islands in terms of ants, their distribution, and their potential impacts on people, on the economy, on agriculture, and so on. Now, to the best of our knowledge, there are no native ants in Hawaii today. I have done a bunch of research into when did ants first arrive and when did people first start talking about ants. The earliest record I could find of anyone using a word for ants in the Hawaiian language was in 1839 when Proverbs in the Bible was translated into Hawaiian. And this is a famous verse in the Bible that talks about ants. And so in the Hawaiian dictionary, we have two words for ants, nao and nona. And by the 1840s, these words were being used in Hawaii. My question that I wanted to know is, like, were people just talking about the ants in the Bible, or were they actually seeing ants on the ground? The earliest reference I have found is this newspaper article from 1864, in which the nao aki was discussed. The nao aki means the biting ant. In reading this article and translating it into English, we see this reference to nao po'onui. It translates to ant with a big head. And I know enough about the ants in Hawaii to know we were talking about this ant, the Phytole megacephala, the African big-headed ant. This is currently the most widespread ant in the islands, and it appears to have been one of, if not the very first ant to become widespread in the islands. The second article with that blurry title of Ka Nao Ho'a, that translates to the fire ant. This is a newspaper article from 1871. This is not the little fire ant. This is the tropical fire ant, Solenopsis geminata. This ant has been around for quite a long time and was making newspaper headlines by the early 1870s. And then there was Mark Twain, who visited the islands in the 1860s. And in one of his letters from home in April 1866, he was complaining about the pests he was encountering here in Hawaii. He complained about the centipedes and cockroaches and fleas and lizards and red ants and scorpions and spiders and mosquitoes and missionaries. So we know that by the 1860s, ants were getting widespread. The first scientific paper published about ants, however, did not come out till 1879. This paper by Frederick Smith purported to talk about 8 species of ants known in Hawaii. I emphasize 8 because he went on to describe 7, and he died a month before this paper was published. So I will forever wonder what was that 8th species he was going to talk about. Starting then, 1879 and onwards, people started publishing more and more about ants. A lot of stuff has been written and published by the Hawaiian Entomological Society in their proceedings. This society was founded in 1905, and we've got a century's worth of incredibly detailed notes about what entomologists were observing throughout the 20th century. Not just ants, of course, but lots and lots of other stuff. And so they would report new records of ants, like somebody saw an ant, they're reporting it, and we've got these incredibly well-documented sometimes anecdotal observations of ants as they arrived. Argentine ants arriving around World War II. Papers emphasizing new island records of old ants. And these papers just go on and on and on. And from this information, we can reconstruct checklists of what ants were present at a given time. This paper by William Morton Wheeler was published in 1935. It's one of the early comprehensive checklists. Then in the 1960s, Huddleston and Fluker, Wilson and Taylor, both pairs of people published papers on the ants of Hawaii. And then in 2005, this very important paper by Paul Kreshelnitsky et al. On the ecology, policy, and management of ants in Hawaii. But not a lot has been published since 2005 in terms of summaries of all the ants that we have. But from these papers, we can reconstruct a timeline of the arrival of ants. And what it shows is that very consistently, starting in the mid-1800s, we have received colonization of a new ant every 2.5 years, very, very steadily. And this continues to this day. Little fire ants, which we all hear about and talk a lot about because it is a very important one of these invasive ants, arrived in 1999. Currently, it is causing serious problems on 4 islands. Hawaii Island, it is very, very widespread. It's causing a lot of discomfort and drama for our friends on the Big Island. On Kauai, Oahu, and Maui, we are working to identify, delimit, contain, and eradicate all the known populations on those islands. Little fire ants, again, arrived in 1999. A few years later, this paper came out, The Little Fire Ants, Its Distribution, Impact, and Control. Now, I mentioned that little fire ants are widespread on the Big Island. That's where they arrived first, and they became widespread pretty quickly. And this paper that came out a few years later, one might say, dang, I wish we'd gotten that paper a little bit earlier. We could have used some forewarning that this ant was on the horizon. And not to throw anybody under the bus, because I know we're all working really hard. We're all overwhelmed with the challenge of invasive species, and we're all learning from the mistakes of the past. But I want to say we did get a lot of forewarning about little fire ants. The threat from little fire ants has been understood since the 1800s. Carlo Emery in 1893, back when LFA was in the genus Tetramorium still. Carlo Emery wrote a list of invasive ants found in the tropics. He wrote that other species will certainly become just as widespread in the future, including the tiny Tetramorium auropunctatum, now known as Wasmannia auropunctata, little fire ant. In 1936, Marion Smith wrote in discussing the ants of Puerto Rico that the locals there claimed that the sting of the little fire ant is dreaded by them even more than that of the Hormiga brava, which is the tropical fire ant. 1941, Herbert Spencer, discussing LFA in Florida, talks about citrus grove workers being driven from their work by these ants. The premiums have to be paid in their wages to get them to go back to work. Same paper, Herbert Spencer pointed out that there is much danger of artificial spread from palm and citrus nurseries of this ant. 1947, Marion Smith says little fire ants are of considerable economic importance. And in 1950, William Creighton wrote that this ant has been carried all over the tropics in both Old and New World. And then 2 years prior to its arrival in Hawaii, Hervé Jordan wrote, little fire ants represent a visible threat to the biological conservation in the whole Pacific region. It endangers the fragile equilibrium of insular ecosystems. So we did have some forewarning. And the lesson learned is that invasive ants are still coming. Little fire ants were the species number 51 to arrive in Hawaii. We have 67 species in Hawaii now. That means 16 species have arrived since little fire ants. These 16 species all arrived since that 1999 arrival of little fire ants. None of these 16 species are ruining the economy in the way little fire ants are capable of doing. But we only know that in hindsight after we found these ants. So what I want to do is raise awareness of the ants that are coming. There are ants waiting in the wings to invade Hawaii. Here are 5 examples of ants that are known to be invasive in other places, some of varying levels of drama that they incur. The ant in the top right, Solenopsis invicta, the red imported ant, the red imported fire ant, is one of the most dangerous invasive ants in the world. But gaining more and more attention is this ant in the bottom middle, the tawny crazy ant. This ant is eating its way through Texas and Florida right now as we speak. It in just the last decade, it has exploded. This ant reaches gargantuan population sizes, so much so that when you treat for them, the ants pile up in drifts inches deep. Look at these pictures of people with handfuls of dead ants. People have to use leaf blowers and brooms to sweep up the dead ants. We do not want this ant in Hawaii. The only way we're going to stop this ant is if it is found immediately upon its arrival. Going back to the red imported fire ant. That's the red imported fire ant on the left, tropical fire ant on the right. Tropical fire ant, once again, has been in Hawaii since the 1800s. Red imported fire ant is not present in Hawaii. But look how similar these two ants are. Under the microscope, these ants are virtually identical. Their behavior is different. The red imported fire ant is much, much meaner and aggressive, more aggressive than the tropical fire ant. So red imported fire ant would be an absolute game changer. The only way we're going to know that red imported fire ant has arrived is if the people looking at them under the microscopes can tell the difference between these two ants and can raise the alarm early. So again, early detection is essential if a rapid response and eradication is to remain a viable option. When little fire ants were found in 1999, we weren't ready. We had not been keeping an eye out for it. The first LFA under a microscope were not identified quickly. The people just hadn't been trained to be on the lookout for this ant. And so by the time a positive ID was made, the ant had already become too widespread on the Big Island to allow for eradication. So early detection did not occur, and a rapid response was not on the table for that island, at least. Again, early detection requires that we identify every ant to species. That is the main point I'm trying to get across. But I also want to emphasize that it's not as hard as you might think. Compared to a lot of taxa groups in Hawaii, invasive ants are relatively easy to identify. And the data collection and data entry, it's time consuming, but it's not overwhelming. I don't want to downplay my skills and speed. I'm really good at this and I'm really fast at it. But other people, you can learn to do this and it can be done. There's no magic to it. There are roughly 15,000 identified species of ants in the world. There's probably another 10,000 species that have yet to be described. So if you have an ant in front of you and you're trying to pick its ID out of a list of 25,000 species on Earth, that does sound a little bit overwhelming. But the fortunate thing for Hawaii is we're only really exposed to the tramp ants of the world. And out of those 25,000 species, only around 300 have ever been found outside of their native range. Those are the tramp ants. Tramp ants are the ones found in non-native places for those species. So we're only looking at a list of about 300 ants. That makes things a little bit easier. And just to emphasize, again, Hawaii has 67 species known today. Hawaii is similar in size to Costa Rica. Costa Rica has about 1,000 species. So Hawaii could certainly fit a lot more ants, but the challenge to entomologists in Costa Rica is orders of magnitude more difficult than what we face here in Hawaii when it comes to identifying ants. These are the 67 species of ants currently known to be in Hawaii. Again, it's not as overwhelming as it might be for other taxa, but we can make it even easier. Let's cut it down to 52 species. These are the 52 species I've personally encountered in my work at Maui Invasive Species Committee. These are 52 species that I'm now familiar with. And I'm unlikely to see the other 15 species. So knowing these 50-ish species gets me quite a ways into what needs to be done. But let's make it even easier. Let's cut it down to these 20 species. These 20 species comprise 95% of the ants that I encounter under my microscope that is collected by the staff at MISC. Getting to know these 20 species, many of which look very different from one another, covers 95% of the work. So again, it's doable. This looking at the 30 most common collected species by MISC. Over about 40,000 detections over the past 4 years. You can see on the left of this graph, 5 species really stand out. Those 5 species cover a significant percentage. So I'm just trying to point out different ways of listing the most common species. These 5 species comprise almost 60% of our detections. So even to start with, if you knew these 5 species, you're well on your way to knowing the ants that come across one's microscope. And we have this great tool. The good folks at the Hawaii Ant Lab have published a dichotomous key to all the ants known in Hawaii. They update it every few years as new ants arrive. With a little bit of practice, it's very doable to learn to identify ants in Hawaii. Now, talking about the ants that MISC has collected, I think it's important to point out that the data we collect is skewed a bit. There's a couple of biases in our surveys. One is certainly a location bias. We're surveying in places where we know there's little fire ants, where there used to be fire ants, where we're worried that there might be little fire ants. And so nearly all of our surveys are in very disturbed areas at low elevations. We also have what I call a singular collection method. We collect almost everything using peanut butter. A lot of ants love peanut butter, but there's a few ants that don't. And some ants that are obligate predators. They only go after live prey. And so they have no interest in skippy, creamy peanut butter. For example, these 10 genera are widespread. They're not particularly noticeable ants because they're mostly hunting in the forest. They don't come into your kitchen. But we rarely catch these ants just because they're after live prey. This map shows a distribution of where MISC has done surveys over the years. Each of these dots represents anywhere from one sample trap to thousands of sample traps. But you can see where we've looked. Those red dots indicate places where at one point or another, we found little fire ants. A lot of those places no longer have little fire ants, but just to give you some perspective. Because some of these places we survey a lot more than others, we also have this heat map that shows on a scale of 1 sample trap to choke traps, how many traps were placed in a given location. And you can see we focus our attention in certain places, and those are places where there once or currently are little fire ants, and we need to have a handle on that. But over the course of our work, we get a really good picture of what's happening on Maui. Still, you can see there are some gaps. We have almost no information from the national park, from leeward East Maui, from the old sugarcane fields, or from the mountains of West Maui. But what we can do with this data is we can plot maps like this. This is a map that shows about 24 species of ants in a particular stream valley. And we can start asking questions and looking for answers as to how these ants are interacting with one another. Ants constantly interact with other species. They fight with each other. They have territorial battles. Some ants are shy. Some ants are aggressive. Some ants affect agriculture. Some ants do not. And the questions that we have about ants will never be answered if we don't have data. And that's what this work that we're doing at Maui Invasive Species Committee allows us to do. Here is a zoomed in version of that map before. I'm going to go back and point one thing out. Of this 24 species at the bottom right, the last ant listed, Phytoli fervens, over 2,000 samples on this map are of that species. And you can, at quick glance at the rest, you see most of those ants are 10, 20, 30, 40 species, sample traps. Phytoli fervens is the dominant ant in this location. Those are the yellow traps in this map. And this makes me ask the questions such as, why is Phytoli fervens so dominant in the lower left part of this map? And why is there such high diversity just a few hundred yards away in the upper right portion of this map? And those red dots represent little fire ants. How are they interacting with these ants? Little fire ants, we know, push out every other ant in their territory. as they expand in populations. Maps like this help us start to elucidate which ants are resisting little fire ants. I think that's useful information to us. Here's another map. We have two species of Phytoli, of large Phytoli, big-headed ants. Phytoli megacephala is a long-time ant in Hawaii. It's widespread on every island. This map shows that the other big-headed ant, Phytoli fervens, sort of displaces it along windward Maui. And I find that very interesting. It hasn't been here as long. Why does it like the wet forest so much more? Is it going to spread more? How is climate change going to affect this distribution? Is one of these ants worse than the other in terms of things like agriculture, the economy, and the way of life of the people who live here? These are questions that we can start asking once we know where these ants are. So again, some of the questions that come up when I start looking at these maps are, one, which of our ants is most resistant to being extirpated by little fire ants? That could be useful. Maybe we can encourage certain ants that battle with LFA. Who knows? After LFA are removed from a site, we end up with a site that has virtually no ants. And so the remaining ants on Maui will recolonize. What is the sequence of events as ants recolonize an area that's been cleared of all ants? This could be very useful if you're, say, in agriculture. I also want to know which ants are encroaching upon our native ecosystems and that are causing problems for our native arthropods. Which of course leads to the question, what are these ants even eating? Ants need to eat something. If they're only eating cockroaches, sure, maybe that's fine, but I doubt that's what they're doing. So let's learn more about what they're eating. How will our current myrmecofauna react to new introductions? Every time a new ant arrives, it inserts itself into the mix of ants and starts fighting with our existing ants. Which of our current ants have stable populations and distributions, and which are still in flux? This question fascinates me because it can take decades for ants to settle into their distribution. And I'll talk more about that in a second. These four ants are four of the most common ants collected on Maui right now. But these ants, none of these four were here 25 years ago. These four ants arrived in the last 25 years and are now some of the most common ants on Maui. This begs the question of, what will the situation be 25 years from now? What will be the most common ants then? And will that be an improvement, or will that be, will things be worse for us? I wish we had snapshots of the ant distribution going back 200 years, but we don't. But that's what we're making right now. Speaking of populations in flux, I like this example because I think it's really, really interesting. It's not from Maui. It's about Argentine ants on the island of Oahu. Maui does have Argentine ants, but what's interesting in the Honolulu example is that they arrived during World War II. They spread throughout much of Honolulu in the 1950s and 60s. This ant was well known as a highly invasive ant, especially in mainland United States. It's the most common ant in California. And as this ant was spreading in Honolulu, alarms were being raised. But then starting in the 70s and the 80s, data kind of disappears. I don't see much more references, many references to it in the literature. In 1993, Gillespie and Reimer wrote that this species has been in Hawaii for 50 years, but it no longer occurs below 600 meters. It's likely that Argentine ants are only successful above about 900 meters in the Hawaiian Islands. This ant extirpated itself from Honolulu and is no longer found there. And that's fascinating. I'd love to learn more about why that is the case. It's my understanding this ant isn't even found anywhere on Oahu anymore. Someone can correct me if I'm wrong, but I believe that is the case. So ants, the point of this, the moral of that whole story is that the distribution of ants changes over time. This ant is an interesting ant. It has yet to be identified. It's recently started becoming more and more common in our samples on Maui. We know it's in the genus Solenopsis. I suspect it's going to prove to be benign more or less, compared to a lot of the other ants, but who knows? We don't even know what it is. This is the current distribution in our samples at MISC. And it'll be interesting to redo this map 5 years from now, 10 years from now. Does this ant spread? Is it going to start being found on other islands? This is important information that we'll only know by collecting samples and making maps of their change over time. Now, no talk from me, of course, would be complete if I didn't talk about von Zweilenberg's Dracula ants. This ant is fascinating, Stigmatoma zweilenbergi. It was described from specimens collected on Oahu in the late 1930s. This ant has only been collected 9 times on planet Earth. And in 2022, I still get chicken skin when I think about it. I found this ant buried in peanut butter in somebody's ant vial. I thought it was a little fire ant. I almost didn't give it a second look. It's the same size, same color as little fire ants. I dug it out of the peanut butter. I started scraping the peanut butter off of it, squirting alcohol on it. And pretty quickly, I noticed this is a strange looking ant. Look, it doesn't even have an eye. And once I got a better look at it, I was able to make a tentative ID and then refer it to some people who are much smarter than me to look at it. And at that time, and then eventually we saw that this is prior to it being found in Hilo on Maui, it had only been collected 8 times before. And even to this day, it's only been found in 5 places in the world. I could talk about this ant for another hour, but let's move on. I want to talk about this ant. This ant was described in the late 1800s and named for our recently deposed queen, Liliuokalani. And Monomorium liliuokalani is widespread in Hawaii. It's been found on Maui, but it seems to have disappeared. We've collected over 2 million ants on Maui, and none of them has been this ant. This is an ant I would be proud to find in my microscope. And why haven't I found it? Who knows? I'll let you know. I'm gonna have a party when I find this ant. If it's not obvious by now, I love doing the work I do. I really love this work. And so I was able to create this word cloud by plugging a word cloud generator directly into my brain. And all these words and phrases popped up. These are the words and phrases that bounce around inside my head when I'm looking at ants. And you too could have this much fun at your job. That's the moral of this whole talk. Okay, to sum up, new ants are chopping at the bit to come into Hawaii. We need to be ready for them, to notice when they come, to have that early detection, to keep rapid response on the table. We need to identify 2 species, as many ants as are seen as humanly possible. And I also want to make it clear that ant identification in Hawaii can be taught, it can be learned, it can be done quickly, it can be done efficiently. You don't have to be an entomologist. It helps, but if you are a bona fide entomologist, you probably have a lot more work on your plate than just invasive ants. If you're interested in learning more about ants, I want to point out 3 really outstanding websites available to the public. There's antmaps.org. This website displays every published record of every species of ant. This is a map of the tawny crazy ant that I mentioned was taking over Texas a little while ago. You can see the green in South America is where it's native. You can see the brown is where it is not native. You can see it is found throughout the Gulf Coast. South Texas, South Florida are very similar climates to Hawaii. They're kind of the bellwether. They tell us what's on their way to Hawaii. Antweb.org. This website is loaded with 2 things: photos of virtually every ant known to science, and it also has links to almost every scientific paper that's relevant to each species. So look up a species and it directs you to all the papers that have been published about that species. The third website is antwiki.org. This is a wiki like Wikipedia, but it's just about ants, and it's written by some of the smartest people around. I use these 3 websites constantly. Almost every day I'm at one or more of these websites. refreshing my memory, or getting more information about the ants I'm finding here on Maui. With that, I want to wrap up and say some thank yous. The work I do is absolutely not possible without the support of the County of Maui. They're very generous in their support of the work MISC does. We also get very generous support from the Hawaii Invasive Species Council. I want to give a shout out to the Hawaii Ant Lab, especially Michelle Montgomery there, who got me started on this whole business of identifying ants. And of course, I could not do the work I do if there weren't dozens and dozens and dozens of people in the field collecting ants and putting them in my freezer. That's a list on the right of just the current people and recent people who've collected ants for me. And that's it. Thank you. Thank you. I appreciate your time.
Wow, mahalo, Monte. That was a fascinating look at how much we can learn when we gather that intelligence. I don't have the phrasing down quite as well as the MISC ant crew. Um, this was super interesting. If anybody has questions, please put them into the Q&A so that Monte may be able to answer them while we are hearing from Trisha. But we will have a little bit of time at the end of this. It's going to be a little longer than an hour. I hope you can stick around. But at the end of this entire session to answer some of the live questions that we're getting. So now we're going to switch gears a little bit and look at how these detection efforts come to life in the field. And sometimes that gets the help of a very special MISC team member. So I want to start introducing Trisha Dillenburg, who is the little fire ant detection dog handler with the Maui Invasive Species Committee. She'll be sharing how scent detection techniques and Freddie the dog are helping to locate these tiny invaders across Maui. Trisha Dillenburg is a graduate of the University of Utah with a degree in molecular biology and chemistry, and with a background in education and conservation volunteering. She's been working with the MISC Little Fire Ant program since 2022 and has been MISC Little Fire Ant Detector Dog Handler since 2023. So I am going to hand it over to you, Trisha.
Thank you, Beth. Hello, everybody. I'm Trisha. Happy to be here. Thank you, Monte, for setting the bar really high for me. Great job. So this is Freddie. I could talk about him all day, so buckle up. So he, sorry, also having the advancement. There we go. So he detects Wasmannia auropunctata, or LFA. Monte spoke a little bit about that, but they're known in other parts of the world as electric ants. They're very small, 1.5 millimeters in length, native to South America, discovered on Maui in 2009, and they thrive in warm, humid, shady habitats. They give painful stings that linger and may cause blisters and rashes, and they form super colonies that increase in density over time, spreading outward and pushing out other species. And they're easily spread by moving intact nests in plants, organic material, and even cars. So we don't want them here. So why do we want to eradicate them after all that? They destroy Maui's ecosystems and harm nesting birds and sea turtles. They jeopardize way of life in Hawaii, farming, hunting, foraging, and recreation, and they're extremely economically costly to live when they become an infestation, and they cause a reduced quality of life. And they can blind pets in infested areas, and there's potential for people and animals to be allergic to them. So what we do to fight LFA, we respond to stinging ant reports. So a lot of the ants that we get that Monte identifies are from ant reports. And then we make early detection efforts to find them in areas where we know they may be brought in from other islands. So we survey, we delimit new infestations and monitor between and after treatments and execute treatment plans specific to each site. And we innovate to make surveys and treatments more efficient and effective, and have developed custom survey equipment and methods, aerial treatments with helicopters and drones. And even with all that, we sometimes miss surviving nests in our surveys. We have hotspots after a series of treatments that allow infestations to persist and remain in treatment for multiple years, maybe 5 to 8 years on average. For some of them, and infested materials are going to continually be brought into Maui. Unfortunately, we're surrounded by islands that have lots more LFA than we do, so it's going to be an increasing problem. Our crew needs support to continue their work to keep up with our current infestations, and we are probably only going to get more. So let's sniff them out. So this is Freddie, the nose. So he is almost 4 years old. He's going to be 4 in December. And he was trained to detect little fire ants by one of the first persons in the world to train a dog to detect Wasmani punctata. So why can we use scent detection dogs in conservation? Because of their acute sense of smell. So their sense of smell is anywhere from 10 to 100,000 times that of a human. And they can smell a target even when mixed with other layers to 1 to 2 parts per trillion. They're always learning, which is an advantage and also a challenge. 10 minutes of searching is equivalent to a 1-hour walk for a dog. So very stimulating, and they love it. They can be motivated by food, play, praise, or affection. They can do passive versus active indications, and you can develop different search types for different environments. Wind, temperature, shade, sun, elevation, and humidity all affect scent in an environment, so you have to be flexible and respond with the dog according to the unique environment in which you're working. And LFA's unique pheromone is Freddie's target scent. So it's their foraging pheromone. So when they're just hanging out and chilling, not being alarmed, so he can find them even when they're, even when it's raining, and other times when maybe they're not foraging for whatever reason, and we don't find them in our samples, Freddie can still find them. His foundation is obedience. So obedience training first, but there are other methods, like people can use their pet dogs to develop scent detection. So pros of working with a dog. He can find ants in the rain anytime, regardless of foraging behaviors. He can detect very small volumes of LFA. He's cute. And working with a dog is rewarding and uplifting for the team. It's reciprocal. The team supports me and the dog kind of is the little mascot cheerleader for everyone. And he can make the team's process of finding LFA more efficient and precise. The cons, I can't cuddle him and I can't treat him like a pet. He's a working animal, and I have to maintain that professional relationship. And sometimes his behavior is hard to interpret. He'll do things that I have to understand, like, is he communicating something to me, or is he responding to something that I'm doing? He's an animal and can have off days just like a human. He's also very stubborn, but most of the time I'm able to leverage that into his work. And also poop. Lots of poop. So we brought him over in May 2024, and he's from Queensland, Australia. So Craig A. Murray is his trainer. And so we had to import him after his training was complete. He worked with Craig for over almost 2 years, learning to detect fire ants. And then I had to go be trained how to maintain his training and to do all the different search types that he's able to do. So what that looked like is we first transitioned him from Craig as his trainer to me. So we just did that by hanging out when we were in lectures, and then playing with him and then taking care of him. And then during that time, we learned foundations of dog psychology, which is remarkably similar to child psychology, obedience training, and got into a little bit of the science of odor detection, which my background in molecular biology kind of fit right into all of that. And then search training. So as a reward, Freddie is play driven. So when he finds a hide or when he detects in the field, his reward is the ball, which is really convenient because I don't have to carry food with me all the time. I can just have a ball for him. And then scent maintenance, so I have to continually train him to maintain his scent picture. And that's one thing that we learned about. And then also all the different types of searches that he can do. He can do broadacre searches where he just runs around. He can search on a leash, off a leash, wall searches. He kind of can do it all. So. And then what happens when he detects? So in the next slide, I'll show you a video where hopefully you can see a little bit of this happening. But what I'm looking for when I'm searching with him is first attitude change. And that's he's smelling something. So that's a similar behavior that you would see in other dogs when they are smelling food or another animal. And then next, I'm looking for OPR, or obvious physical reaction, and that's his body language shift that happens when he detects LFA. So that's when he starts getting really focused and also really excited, because he knows if he can pinpoint it, he's going to get the ball, he's going to get rewarded. And then indication, so he does a passive indication where he lays down with his nose pointing toward the target, and that's him telling me this is where they are. So hopefully that'll help you kind of catch that in this next video. It happens really fast. So there's his indication. All right, so then our journey to Maui, we imported him from Australia to Maui, and then we transitioned him from lab-produced scent materials that had been produced by Hawaii Ant Lab to live ants in the wild. So we took him to a known site of LFA and worked with him there, because it had been the first time that he'd worked detecting live LFA. And then he detected live LFA in the field after 2.5 days of working him. And this next video will show that also. So this was his first ever live ant indication on Maui. Okay, and then we've had him about 1.5 years that we've been working with him. So we are currently raising our own live LFA to produce materials for him, for his ongoing training. And we also use those live ants from the colony to do live ant tests, so that we can just give him a variety of training. And I've used that to increase his volume threshold, because at first he showed aversive behavior to large volumes of LFA. Even if they were isolated, he wouldn't go near it. And so I had to work up to doing small volumes of live LFA, all the way up to now he will detect a whole intact nest with thousands of LFA, and he no longer shows aversion to it. So that we're hoping will translate to him being able to be used for other isolated pockets of high volumes, like in cargo areas or nurseries. And then since then, he's helped us discover 3 sites of LFA and helped to declare 1 site eradicated. And he's also been recently finding hotspots for us. And so how do we collect the data of his searches? So we have some maps coming up. And so just to show what you're going to see, we look for the question mark, which is the attitude change I described. And then OPR is a exclamation mark. And then an indication is a finger point. So this is a map that shows, and it kind of like the perfect example of how we used him for delimiting a new site that we discovered this summer. So we decided that we would do a loose survey and mark where he started indicating, and change to a higher volume survey, so a tighter survey. And so I was working from the left to the right. So the purple line is my track, and then the blue dots are sample vials. And the blue means that there were no ants or non-LFA, and then the red dots mean that there were LFA. And if you see, this is where the exclamation mark is where I was standing when he first showed OPR, and then the finger point is his indication. And if you notice, that's where I was standing. The red dot is where he hit the first spot of LFA, and you can see we were walking right into it, and he was correct. So this map is like a perfect example of how he's worked in the field for us. And then this is an example of a stream survey, just to show kind of some of the area he can cover. So we started and worked upstream, so at the upper right corner, and then we worked upstream and all over the banks of the stream to the other side. And just, I put the line there to show 50 meters, to show how much, that was probably about a 10-minute survey, and how much area we were able to cover. So aside from taking him in the field, every day I have to do maintenance and obedience with him. Like I said, his foundation is obedience training. So, and I also have constant scent picture maintenance training. Ideally, we like to use 50% lab produced materials and 50% live ants, and then time spent 50% training, 50% field time. So scent discrimination is something that I do with him so that he can filter out other scents, like common things that he would encounter in the field, or even contamination of me. So I'll touch objects and hide them, and then put live live ants or scent material for him to find, and he has to go through everything, even peanut butter. He'll go through it and he can't indicate it or play with it or eat anything and just focus on finding the ants. And he does really well with that. And then his obedience foundation makes him really reliable and trustworthy in the field, which is why Craig's method is to train dogs obedience first. So that means he's not distracted by birds, cats, other dogs, or people. So this video here, I hope the sound will come through, but this is an example of I was doing a training with him in the field on live ants, and he was searching and a flock of nene landed and they were alarmed and freaking out, and it didn't faze him at all. So. And then this is an example of scent discrimination. So there's all these different objects, plastics, metals, and then there are hides. And he's got to sort through that and find the LFA. And he's really quick. So, all right, so our successes. Our success is that we have a strong dog handler relationship. Freddie and I are really in tune with one another, and he's become like an LFA compass. So I'm constantly practicing interpreting his behavior, but he's already shown us in the field how he can tell us where we need to look and focusing on areas that we might otherwise have missed. And he's also found ways to communicate when we were surrounded by LFA completely. I've noticed it was a weird behavior when I accidentally took him too far into an LFA infestation where he stopped searching himself and came to me and initiated play, which he had never done before. And then after I looked at the data, I realized I put him way too far into an area that we were saturated with LFA. So he didn't know what to do. So that's what he did. And he did it another time when I was responding to an ant report, and I saw that same behavior and it reminded me. And after our survey, sure enough, we were surrounded by LFA. So that was really cool that he has found his own way to tell me when he doesn't know where to indicate because they're everywhere. And then also, like I mentioned, I train him to detect live ants in a variety of volumes and search types. And then we also, at the beginning, we weren't sure how useful he could be on treated sites because we thought, well, maybe ants that have been treated with insect growth regulator may smell different to him or he was going to respond differently. But since then, we have used him on treated ants in the field, and he has been able to detect hotspots of IGR treated LFA. So that question of will he detect IGR treated LFA? Yes, he will. And obedience and training maintenance is ongoing, but overall he's very consistent. He's kind of like the perfect dog, really. I think I might be a little biased, but he also really loves his job. He's visibly happy and exuberant all the time and loves to work, whether we're training or in the field. He's always motivated. It's really easy to get him to do what I need him to do. So he's a lot of fun to work with. And so here's an example of the IGR. These were the first ones that he sniffed out for us. So we did a search and I was zigzagging this way and he indicated in 2 spots, and a subsequent survey turned up that there was a hot spot there. And then some challenges and questions. So keeping a live LFA colony was a huge challenge when we realized we were going to have to start producing our own material and we needed a live LFA colony to be able to do that. But we've done it, and we still have a live ant colony that we've been able to use to supplement his training. And then also sometimes it's really hard to confirm his indications, but that's exactly what he's for, though, is to find LFA in ways when our traditional methods can't. So reading him correctly is vital, and then trying to figure out what to do with the data that he produces after search is kind of ongoing. It's dynamic. And then designing data collection to accurately capture Freddie's search responses. We're getting better and better, but that's also ongoing, and it's new. It's all new. Obedience and training maintenance is ongoing and must constantly pivot to address behavior issues. So if something happens that's unpredictable, I always have to address any discipline and respond to his behavior at the moment. So it forces me to have to be very flexible with him. And then another challenge is that not all environments are suitable for Freddie to survey. Properties with loose dogs are always a no. Tops of cliffs, because he goes where the ball goes. So controlling the ball is vital. Large boulder piles, because again, when he's searching, he'll go anywhere. He's tenacious. He's not looking out for his own safety. I have to do that. Tall vegetation is a challenge because I have to read his behavior. If I can't read his body language, if I can't see him, then it's pointless to take him there. And then like I said, his safety in the field is a constant focus of mine. I have to keep both myself and him safe. And then he's also detected dead LFA. So that raises the question, how long will an infestation trigger a response from him after we've killed all the LFA? So that's another one of those hanging questions that hopefully we'll answer in the future. And then moving forward, we're going to continue to expand his training and continue to build trust, consistency, and reliability with his searches, and fine-tune data collection systems and field search methods, and then further incorporate dog searches and ant report follow-up, early detection, post-treatment monitoring, delimitation, nurseries on other islands, and port of entry surveys. We're hoping to go to Kauai to work with HISC soon. So that will be his first off-island trip, and we're looking forward to doing that. So some quick FAQs. How much area can he search at a time? I usually search him about 10 minutes at a time, and that will vary based on the environment. And how long can he work? So the first dog that Craig, it was a Labrador that Craig taught to detect little fire ants, worked until it was 9 years old, and Freddie's currently 4. So we're hoping to get another five years or more out of him. Can he play with other dogs? No, he can't. He has to be completely isolated at all times, but he's already got some play dates for his future when he retires. And then what does he do when he's not working? So I am his 24/7 caretaker. And when he's not working, he's at home chilling in his kennel. It's actually the only time when he's not being stimulated, because if he's with me, he's completely focused on me, and it's not rest for him. So when he's not working, he's resting at home. And why can't he be cuddled or treated like a pet? And that's because he hasn't learned yet to look to humans as a source of affection. I can use affection in his training, but if he knows that he can just get affection for being cute, which I don't think it would take him very long to figure out, it could ruin his motivation. So we want to keep him motivated to work, because otherwise his reward could just be being with me, and he might not want to search anymore, and that would be very difficult to train out of him. It would not be good for him. And does he get stung by LFA in the field? So he probably has been stung by LFA, but LFA aren't actually aggressive like RIFA or TFA, and he's furry. And normally we're not taking him really far into an LFA infestation where he would get stung. And he's smart. Most dogs don't want to get stung, just like field workers don't want to get stung. So he will learn like a safe distance where he can indicate where he's not going to get them on his face. So I don't think he's been stung recently. And then does he ever refuse to work? He's never refused to work. Usually he's raring to go. And is he ever wrong? So he does indicate sometimes, which we call, Craig's word for it is a goose, because that's his way of getting my attention. And so it's his way of being like, hey, there's something over here. And so it's like an indication where he's not showing OPR. So I'm usually already skeptical of it. So we have a way to track that in our data also. And it's an attention seeking behavior that has to be, it's part of what I have to keep up in my training with him. So, but he's not really wrong. It's just a matter of, am I interpreting him correctly? All right, so send us your ants, and if they're LFA, you might get a visit from Freddie. So, and thank you to the county and everyone who has supported this project. And that's all I've got for you. So thank you.
Wow, mahalo. That was an amazing look at how Freddie is changing the way MISC is finding and responding to little fire ants on Maui, here on Maui. And I was especially fascinated about how you are able to translate not only how you work with Freddie, but how you're able to translate those surveys into data that then can inform how overall little fire ant is worked with on Maui. Super interesting. Now, I want to open it up for a quick Q&A for both Monte and Trisha. If you have a question, drop it in the Q&A box or raise your hand, and Chuck will actually help with answering questions if anybody has any. And it is okay. They're also open to questions if you want to reach out, go Google MISC, look at the little fire ants. You can find these guys. But we'll just see for just another moment if anybody has any questions. And while people are thinking about that, I not only want to mahalo Trisha and Monte for taking the time to give this presentation and allowing us to record it so we can share that on YouTube so that people can find this information and learn from it. I'm sure they'll find it just as fascinating as I do. So this will be shared also out on the HISC YouTube. If you want to share this with other folks, look up our HISC support YouTube and you will find this presentation so that you can share that with others. And Chuck, do we have any questions that have come up?
There was one comment from Brooke Mankin about wanting to see the full Monty, a whole hour-long presentation from him. And I said that they could make arrangements in private for that. But for now, we don't have any other questions. So I wanted to ask one for Trisha. You may have answered this. I'm sorry if I missed it, but what's Freddie's work week like? How many days a week does he work?
That's a great question. I try to work him every day. So I do shorter days with him so that I'm not working with him in the heat. So we'll work usually in the mornings, whether I do a field day or do training at the base order or a combination of both. But he works Monday through Friday. So, and sometimes he'll do demonstrations too. So he likes to show off. So.
Great. And I still don't see any other questions in there. So I did have one other one. And you may have answered this as well. Sorry if I missed it. Did the different populations of LFA in Hawaii have any different scent signature, or do they differ from other areas where they've been moved around the world, or are they essentially all one odor that Freddie can detect?
So the theory was that the ants that he'd been raised on, so those were from colony raised ants from Hawaii Ant Lab that produced his scent materials that he was trained on from when he was a puppy, were going to probably smell slightly different than ants in the wild in Maui. And there is a theory also that ants populations, if they've been separated, so a population of LFA in Ka'eleku might have a very slightly different smell from a population of LFA in O'ili, but that their overall signature pheromone, their fingerprint, I guess, would be the same, but slightly different, which is part of why his training includes having ants, like in our colony boxes, we actually keep the ants separate based on whether we took them from O'ili or Ka'eleku. And then once we separate them, they don't get mixed. And then we still are working him both in the field and also training him in the field because we do think that theoretically they probably do smell a little bit different based on their environment and isolation. So did that answer your question?
Yes, it did. Thank you very much.
Well, mahalo to everyone for joining us today. Happy Halloween, and thank you for joining us for the closeout of Stop the Ant Month. I wanted to share with you if you want to share this presentation with others, it is on the HISC YouTube there. You can copy out that link and share that out. And I guess we're going to wrap it up now. So mahalo for joining us. Mahalo to Trisha and Monte. And happy Halloween, everyone. Aloha.